I guess to get started, I'll pose this to anybody who wants to jump in. We talked a lot about the role of standards, but how do you see those standards supporting regulatory applications and advancing the field? The right button.
The person, yes.
Okay, so you guys are staring at me. All right, so how, say that again, standards.
Yeah. So how do these discussions around standards enable regulatory applications of complex in vitro models?
I mean, I think that's I think it's important because I don't want to say we're going to have an OECD, type of validated guidance, test guidelines for all CIVMs. Of course, that would take many years and a lot of work to do. However, we need something. We can't. We need to have some type of standards to know that if someone's using NAM X in their lab to assess something, and then someone's using the same one somewhere else. We need something to have, again, from the regulator side. We're relying on the data that's in front of us is adequate. And then, we need to be able to have confidence in those data. So, having some type of set of standards that the assay can be run through, that I know when I'm reading those data and I'm looking at those data, I don't have to question the validity and the confidence in those data. So how we get there, I'm leaving that to you, to other people. To inform me when I'm reading that study report and the data, that I don't have to question the validity and the confidence.
Yeah, I just want to add, even though I work at NIST, not at FDA, in my conversations, with other people from FDA, it's important. Well, it makes a bit less. It makes it a bit faster for reviewers, I think, when they have a standard that is referred to when they are reviewing, so that they don't have to go through a lot of data to prove that is true. There is something that is said, they give their results, and I think is I mean, I'm simplifying it. I'm quite sure it's not that easy, but it's a lot less data to look at for the reviewers, so things could move, in a faster way with less effort. And overall, I just want to mention too, so this also help the smaller companies even more, because they maybe don't have all the resources when they're trying to build up and grow, when they want to get something out. So they would need to use a lot of their capital to also work towards that the development. So that will help in democratizing more this getting this out of the doors from big and small companies.
In terms of what we see in the public-private partnership approach, standards approached in a way that allow people to fully understand how to report consistently what they've done, not telling them what to do, is the way to approach standards. Because there's always this, whenever you have entered into a gazillion types of conversations about standardization over the almost 20 years of our existence, for clinical data, for clinical biomarkers, for model-informed drug development tools, for clinical outcome assessment tools, and some of the first reactions you get when you introduce the topic for the first time is, that's going to stifle innovation. Not true. If you focus the standards on making sure that everybody reports reliably what is it that was done, that helps inform exactly what Amy was describing, and that helps then narrow down the evidentiary considerations. It's not about stifling innovation and telling people what to do, but making sure that everybody's reporting what they are doing in a consistent and reliable manner. That has been our consistent experience.
I'll give anybody online the opportunity to chime in now.
Yeah, maybe I'll say just a quick word on maybe from the technical standards point of view. So if I take, for examples, the examples that I made and also Darwin made on materials, for instance, and the absorption of these materials. So I guess that the emulate people will have a very stable protocol to measure the absorption of molecules into their devices. However, if we want to facilitate the whole industrial ecosystem around these, the standard on a material absorption measurement will also help other companies in providing the same data in the same way. And then when this data comes to the regulators, they can be reassured that this, let's say, technical issue has been somehow solved in a way that is agreed by the international community. For instance, also another example that comes to my mind when we think about the bubbles that are usually coming in microfluidics devices. For instance, if we have a clear and common way of making sure that these organ-on-chip are bubble-free, and that all organ-on-chip developers can use the same standard to demonstrate that there are no bubbles or there are ways to get rid of these bubbles, then these could, ensure the reliability of the device, but also of the data that it produces. So these are just two practical examples that come to my mind.
Ivan, do you have a question or comment?
Yeah, Ivan Rusin, Texas A&M. I'd like to thank the speakers for really presenting two sides of the coin. One is how to really prove that the new complex model works. And then the other side of the coin is how to standardize something. So to me, it seems to be the horse and the cart type of a situation where maybe putting standards before the horse, showing that you actually have a model that works or addresses the need before you start standardizing things. So maybe, our proponents of standardization can actually talk about how do we actually standardize something that actually works.
Yeah, so it gets back to our experience with that's a great point that we have heard, or I have heard over 20 years at least 250 times. So that's top of mind for these types of discussions. Once you identify a tangible unmet need in drug development that can be solved through a CIVM, first next step is to define a context of use. The context of use is going to dictate what kind of data you need and what kind of evidence you need to show with such data. That is going to then show you the kind of pieces of information that you need to put in front of regulatory agencies for the qualification reviews. That gets to the point that Amy raised and the point that was raised by the other regulatory agencies, and that has been raised countless times. Once you start with that notion, that's when you start honing into the reliability of the data that needs to be put in front of regulators. And that's when you start talking about the need for any standard that already exists. There are some standards that already exist that you don't need to reinvent. But the potential for, once you envision this solution being adopted, how do you ensure that it is reliably adopted? And that's the answer to the cart before the horse question that you asked, which is a very important one, because then that shows how when the tire hits the road and the solution starts being adopted, how people need to make sure that they report reliably what they're doing, not necessarily telling them, like I said in the beginning, what to do, because it's all governed by the context of use. That has been our experience, and I think that's the way I would propose that that be embraced in this conversation. And I'll leave it at that for now.
If I may, yeah, I think it is a very important point to note that we have to understand how we want to use these models, define your context of use appropriately, and then show that, you know, it is actually giving us a useful answer. I said, and then once you know that it's giving you a useful answer, I think that's where you can, you know, increase the rigor and the standardization aspect of it. But in terms of standardizing, I see, you know, a more immediate help where it can ensure that we've got, you know, good reproducibility in the generation of the models themselves. So irrespective of the context of use and how you actually use the model, but creating standards around how the model is actually created, I think could help with some of the reproducibility challenges that we have seen in the field over the years. And that would give us, you know, a really good foundation from which to start with all of our COU testing and, you know, then the qualification and validation aspects of things.
Yeah. Jay Hickman, Hesperos, University of Central Florida. Actually, again, I agree completely with Ivan, which is a very unique experience. But my question is really directed at Sonia, because she said that the EMA is trying to remain very flexible in terms of what types of systems that they would evaluate and things like that. But yet ECVAM is putting out very rigid standardization protocols right at the moment. So how does the EMA sort of deal with that issue?
Okay. Thank you for the question, Jay. So the flexibility we intend to have is to write qualification criteria that will allow for different methods to be used. So independent of the type of cells, platform, etc., you have to answer the questions whether the method is usable in that context of use. The standardization program that Monica describes is seen a bit as independent from that, if that is what you mean, or if you are referring to the more formal validation criteria that are currently out there and that are needed more to get a method towards OECD test guideline approval. That's not clear to me.
We're pretty much looking at it in general because yes, there's specific, but and there's a whole issue of efficacy versus safety, which we'll get into in the afternoon, where, you know, I can understand for safety there is in trying to get it validated as a drug development tool. Some of the things that are being developed that ECVAM is looking at might be more applicable and along the lines of what you're talking about. But then there's for efficacy, where you don't necessarily need that rigidity, okay, in the models, okay, and how do you deal with that? Because it's generally you don't put out, at least I've not seen it so much from the FDA or the EMA, putting out different standards for efficacy versus for safety. Yet they are two totally different, okay, in terms of what their criteria are. And so by setting up standards, okay, that are one size fits all, okay, I think you'll potentially miss a lot of innovation in the field is what I'm kind of really driving at.
Hi, I mean, I think I want to point out that keep in mind, I mean, from my perspective, the level of qualification and or validation is completely going to be dependent on the context of use. So you gave the example, I'll go all the way to the other end, like drug screening. If an assay is going to be used for screening purposes to take, 100 compounds and run it through and pick the candidate one, the level of validation qualification you need for that is much less than if you're going to be using a NAM for safety to replace an existing safety measure. So I don't think there is a one size fits all for the standardization. It depends on the context of use, and that is going to dictate the amount of validation standards. Does that make sense?
But that kind of what people do a lot in terms of when evaluating MPS is that people looking at safety will go always to the most severe thing that we could probably reproduce, which is systemic tox or something else like that. And you just gave an example, which is, you know, really kind of pretty much far the other way. Most of the people are working in the middle. Okay. And what we're trying to do is, I'm trying to do as president-elect of the new NPS society, figuring out how all the people in the middle can deal with the regulatory aspects.
Yeah, I mean, I think the key word that people are going to say today is, you know, context of use, whatever it is, that is really going to help guide how much you need.
And if I may just introduce a perspective there, because it is 1,000% right. It's also make the distinction of evidentiary considerations for a context of use for a potential qualification versus standardization. Those are two very different things. The question I think that Graham asked in the beginning was regarding how the level of standardization for reproducibility is that's one thing. Now, the bar of evidence for a specific qualification is dependent on the context of use statements. And when you start entertaining, this is our experience, I'm not speaking for regulatory agencies, but this is our experience in working with the regulatory agencies in presenting to them documentation for potential qualification. The common denominator that we have seen is that when context of use statements for completely novel drug development tools for which there might not be a precedent are discussed openly, and then that level of innovation is factored into the discussion about the evidence to support a given context of use. Once you start building qualification after qualification, you may see that pop up in guidance documents or in communications about, hey, so the bar for this specific context of use starts looking like this. That's a different kind of standardization. I just want to make sure that we don't confuse the different levels of standardization, because then that's a distinction.
Right. Very few
I
people make that distinction is the point I'm trying to make. Okay. Thank you for your discussion. This is great.
Do the online participants want to chime in now?
Yeah, maybe just indeed confirm what has been said. The context of use will drive what is needed for qualification and whether a full-blown qualification will be needed. Because if we're looking at efficacy models that are used right now, the evidence that is provided for efficacy comes from very diverse models, in vitro as well as in vivo models, which are not necessarily qualified for that context of use, because this is very much research based and there are no specific as per guideline recommendations for primary pharmacology. So in that sense, a lot is possible already right now. If you have data, you can submit them, even if they are in a complex in vitro model that we have never seen before. But of course, this has to be designed and used that you as a method developer or as an end user have confidence in the results that they are producing. Whether this needs to be standardized, but at least it needs to be characterized appropriately. Now, if we're talking about safety, if you are going to use your safety model to replace really a risk assessment in an in vivo model, the requirements will be much more stringent than if you are using it as a pieces of a puzzle to construct a weight of evidence. But we cannot give you an absolute black or white answer at this point in time. It really depends on what your intention is to do with that model within the drug development context. What kind of decision are you going to take with it? And then you will have very flexible requirements. But this is all in a dialogue with the regulators that you have to discuss this. And we are trying to put bits and pieces together and to give you the qualification criteria, but we will not be able to make qualification criteria for each and every context of use. That's just unfeasible. So that's what I wanted to add to the conversation. Thanks.
Yes, I just going back to the question, right? I just want to mention, right, that in my experience, when it's a new field, people feel like the standards are coming too early. And this happened in is microfluidics, and these fields are really complex system. It's not like the electrical systems that you can put a standard for X, Y, and Z, and it is less complex. But having said that, and I know that this crowd here is mainly about context of use and all that. There are other things that have to be also looked at. Like, for example, as I see it coming to these workshops and panels, is like you have something like 3 different things that you could think of. First is, what are the materials that you're going to be using, like the biomaterials, right? Like cells, media, as Jay mentioned at the MPS, the ECM, right? So that having a standard of some sort that will allow, first of all, everyone to know that when they're buying from ATCC, let's say, or some other company, what they're saying it is that cell, that's important. The other part that I see is the engineering part of it. So there are a number of things, like, as I mentioned, flow, shear stress that is not maybe taken into account, and having some sort of way that you could say what I'm putting these cells through about this in particular is what it should be, and everybody should really say, if I have X shear stress, the cell should behave in Y way, right? And then you get the concept of use, which is what we're talking about here, which is a bit more complicated too, because everybody have their own system. We're not looking into what everybody's putting in. We just try to put in something and see what happens when it comes out. So I just wanted to mention that it is very common for new technology to have this, I'm going to call it a bit of a pushback. And this is not the only field that have seen that. But I think there are ways to really try to move forward some of these things that are going to be important along the way. So I just want to add that.
I think there was a question online.
Yep, we do have a few remote questions, a few from Tina Morrison, actually. The first is for Amy. Are there any opportunities to adopt the WHO concept of reliance for adopting NAMs from different jurisdictions?
Can you actually, I didn't repeat that question from what?
Are there opportunities to adopt the WHO concept of reliance for
Like
adopting?
WHO. Oh,
Yes.
Okay. I'm looking at Nikisa. I don't know the answer. Not that I'm aware of.
Yeah, I'm not sure about what they're referring to with respect to the WHO. Can they clarify what that means?
We'll bring her up. Let's see. She's trying to talk. Okay, she should be able to talk now.
Hi, good afternoon. Can you guys hear me?
Yes.
Yeah, thanks for letting me respond to that. So there was a report that came out from the WHO, I believe in late 2022, that CDRH is adopting. It's an extensive document on the concept of reliance and being able to borrow from regulatory bodies, different decisions that are made about medical products. But in this case, it would be about qualification. And I wonder if there's an opportunity to bring leaders together from the different regulatory bodies interested in advancing alternative methods to have a discussion about reliance and qualification for regulatory purposes in this context.
So, thank you for explaining that, because when you said CDRH, that sort of made things a little bit, easier for me to understand. CDRH really does rely on standardized test methods. They're the sort of paradigm used by them for evaluating the devices that they evaluate is very different from that of CEDAR. CEDAR doesn't use any standardized test methods from what I know. And so I think that what we're talking about in terms of standardization here is a little bit different from how CDRH addresses standardization, like a standardized test method. And so I think that, it's important to not kind of think about those things as being equivalent. I mean, I think here what we're trying to say is that trying to apply regulatory, get some sort of regulatory, significance out of these methods requires very specific approaches for the context of use that we're talking about. Whereas when you're standardizing, you're trying to standardize something that's been made very generic and it's generalizable. And so that is, we can't really do that right now. I don't think we can ever do it really, by developing standards that at the level that we're trying to, or people are talking about. Sometimes when you're dealing with a very specific method for a very focused application, you just have to standardize it for the respect, to the extent that you can actually depend on that data to be able to interpret it correctly. So, in my opinion, that is going to differ every time you use that method, potentially, even in a drug development application, depending on the sort of indication or how that drug is going to be used. And so there are going to be differences there. And so anyway, the point I'm trying to make is very hard to be able to say that you can develop standardization approaches at this stage of when we don't even know how we're going to use these tools.
Right, but again, if I may, just draw back to the distinction I was bringing up before. It's so standards for reproducibility are one thing. Standards for what bar of evidence, it's going to be dependent on the context of use, and if there's not a first context of use for one specific application, that will be factored in inside experience that when we talk to FDA, when we talk to EMA, when we talk to PMDA, that gets factored in to the conversation. But I think Tina's point was, maybe I understood it a little bit differently. And thanks for putting that into context, Tina, for the WHO document for reliance across regulators for devices. And so reliance on what the different agencies are looking at in terms of submissions with specific context of use for tools like CIVMs. Our experience has been in all the other areas, biomarkers, COAs, quantitative drug development tools, etc., is that we always tend to present the same documentation and the same context of use to all agencies, not necessarily in single joint review processes, even though that's sometimes a possibility, but we present the exact same thing. And in those documents, there's always a section that we include in any CPAP document that summarizes the entire regulatory history of the conversations that we've had with FDA, EMA, and PMDA for every document to any agency, in the hopes that helps put everything in a context, I think, to the point that you were raising, Tina. So that would be my take on interpreting what you raised, but you correct me if I'm off base there.
No, I think that's correct. And to be clear, the WHO document is not specific to medical devices. I mentioned medical devices because they're the ones who are adopting the concept around reliance. And what I will say, while many medical devices are fundamentally physical objects with, electrical or mechanical or physical components that and that can, in some cases, allow for standardized technical test methods to be developed and adopted. I do believe there are a lot of synergies that can be borrowed in this space, particularly around the organs on a chips and the microphysiological systems, as demonstrated by some of the characteristics that Dr. Reyes presented. And having had experience in this, I'm happy to talk with other folks about this conversation at another time. But I would encourage to open the dialogue around borrowing qualification of tools from different regulatory bodies as a first step, figuring out how we can have that conversation as well.
Yeah, I just want to point out one thing, I think, from my presentation, just to keep in the back of your mind, is that, if, an alternative method is quote, accepted by one regulatory agency, even, it does not necessarily mean it's going to be accepted by others. So, it's clear in their difference, and even in these ICH international guidances, there are distinctions between the different regulatory agencies, even though this is a global document.
Sonia, do you want to?
Yeah, indeed. I think indeed, I had it in my talk. International harmonization is really important, and there are differences, of course, but there are also a lot of tools to try and harmonize. I talked about the joint qualification advices between multiple international regulatory agencies. That's one. But the other one is the very recently set up International Medicines Regulators Working Group on 3Rs that really has the intention to kind of exchange experiences between regulatory authorities around the globe. And you have seen there are many already included specifically to tackle these kinds of issues and to try to avoid them. Will we be able to avoid them completely? I'm not sure, but at least we can try to converge as good as possible to a common position that we will not have this big discrepancy at the end of the day between the regions. Thank you.